Good evening, students. Today we'll be dealing with the histology of the cerebellum. It is located in posterior cranial fossa. So there is superficial cerebellar cortex, which is thrown into folia and fissures, and there is a core of white matter, and there are deep cerebellar nuclei, namely dentate nucleus, emboliform nucleus, lobose nucleus, and fastigial nucleus. Cortex is thrown into folia and fissures. Each folium is having superficial gray matter and central core of white matter. You can see this is the cerebellar cortex, which is thrown into folia and fissures. So this is the folia separated by fissures. You can see here this is the peripheral gray matter, which is the cerebellar cortex, and you can see the core of white matter in each folium. So and this is the white matter, and these are the deep cerebellar nuclei, namely the dentate nucleus, globose nucleus, emboliform nucleus, and fastigial nucleus. Cerebellar cortex is having three layers, namely the molecular layer, which is the outer layer, and it is having basket cells and stellate cells. The second layer is the Purkinje layer. It is the middle layer having Purkinje cells. And the third layer is the granular layer, inner layer, having granule cells and Golgi cells. So you can see here, this is the outer molecular layer with basket and stellate cells. This is the middle Purkinje cell layer with the cell bodies of the Purkinje cells. And this is the inner granular layer, which has maximum number of cells. The main variety of cells in this is the granule cells and the Golgi cells. And you can see this is the central core of white matter in the folium. Molecular layer, it is a superficial layer which lies immediately below the pia matter. Contents, if you see the fibers, these are unmyelinated nerve fibers, nothing but the parallel fibers of granule cells, sensory climbing fibers, and dendrites of Purkinje cells and Golgi cells. The cells are basket cells and stellate cells. It stains lightly due to fewer cells. Purkinje cell layer is having the flash-shaped cell bodies of the Purkinje cells. You can see here, because the dendrites are entering into molecular layer, axons will pass through the granular layer into the white matter of the cerebellum. Granular layer, innermost layer, contains of cell bodies and dendrites of granule cells. Then Golgi cells, brush cells, which are the glutamatergic interneurons in the cerebellar cortex. Sensory mossy fibers with synaptic glomeruli, and this layer is deep staining because of more number of cells. Purkinje cells, these are flash-shaped cells. Their cell bodies are present in the Purkinje layer, whereas dendrites will reach the molecular layer. Axons will pass through granular layer into white matter to reach the deep cerebellar nuclei. So the proximal unmyelinated part of axon synapses with the collaterals of the basket cells. Recurrent axon collaterals of Purkinje cells synapse with stellate, basket, Golgi, and other Purkinje cells. Input is from the granule cells, basket cells, stellate cells, climbing fibers, and recurrent axon collaterals of the Purkinje cells. Whereas output is given to deep cerebellar nuclei, lateral vestibular nucleus, stellate nucleus, basket cells, Golgi cells, and other Purkinje cells. So these are the sole output neurons from cerebellar cortex, which is inhibitory. Granule cells, they are very small. They have round cell body. These are excitatory neurons. So some in the granular layer, if you see, there are cerebellar islands or glomeruli, which is formed by the mossy fibers with 10 to 15 granule cells and Golgi cells. So they are located in granular layer. Axons enter molecular layer and form parallel fibers and cover a territory of 500 Purkinje cells. Input is from the mossy fibers, collaterals of climbing fibers, recurrent collaterals of Purkinje cells and Golgi cells. Output is to the Purkinje cells, stellate cells, basket cells, and Golgi cells. Stellate cells, these are located in molecular layer in the superficial part. Dendrite synapse with granule cells, climbing fibers, and axon synapse with dendrites of Purkinje cells. They are inhibitory neurons. Basket cells, these are located in deep part of molecular layer. Dendrite synapse with parallel fibers of granule cells, recurrent collaterals from Purkinje cells and climbing fibers. Axons cover a territory of 10 to 15 Purkinje cells. Axon collaterals form baskets around cell bodies of the Purkinje cells. They are inhibitory neurons. Golgi cells, these are located in granular layer. These are inhibitory neurons. Dendrite synapse in the molecular layer with the climbing fibers, recurrent axon collaterals of Purkinje cells, parallel fibers of granule cells. In granular layer, they synapse with the mossy fibers. Excitatory input is from mossy fibers and parallel fibers of granule cells, whereas inhibitory input is from recurrent axon collaterals of the Purkinje cells. Output is to the dendrites of granule cells. They reduce excitability of the granule cells. So, what are the afferent fibers reaching the cerebellar cortex? There are mainly two types, mossy fibers and climbing fibers. Entire afferents to the cerebellar cortex is mossy fibers, except the one which comes from the inferior olivary nucleus. So that's what I have written here. All fibers entering cerebellum are mossy fibers except olivocerebellar. So they synapse with many granule and few Golgi cells. Origin is from vestibular nuclei, pontine nuclei, and spinal cord. One mossy fiber will terminate into 30 to 40 rosettes, which form central component of glomeruli. So one rosette is surrounded by 15 granule cells. One mossy fiber excites 450 granule cells, which through parallel fibers excite 450 to 500 Purkinje cells. So the pathway to the Purkinje cells through mossy fibers is there is an intermediate cell called granule cell. Mossy fiber, granule cell, and Purkinje cell. Whereas if you see the climbing fibers, the origin is from inferior olivary complex, pass through granular and Purkinje layers to reach molecular layer. One climbing fiber influences one Purkinje cell. So it gives collaterals to deep cerebellar nuclei in molecular layer synapses with stellate and basket cells. So efferent fibers from cerebellar cortex, sole output is from Purkinje axons to the deep cerebellar nuclei and vestibular nuclei. Inhibitory to the cerebellar nuclei, fibers from dentate, globose and emboliform leave through superior cerebellar peduncle, whereas fibers from fastigial leave through inferior cerebellar peduncle. So glomerulus, it is located in granular layer. It is covered by Birchman glial cells. So these are specialized unipolar cells with cell bodies in Purkinje layer and radiating fibers to molecular layer. A rosette of mossy fiber is present in the center as presynaptic element. So there are claw-like dendritic expansions of granule cells, which is postsynaptic element. Axon terminals of Golgi cells synapsing with dendrite of granule cells, like presynaptic element. You can see here. So these are the nuclei of the granule cells and Golgi cells, where these empty spaces are there. So these empty spaces are called as cerebellar islands or glomeruli within the granular layer. So this is the Purkinje layer and this is the molecular layer. So now you can see here, you can see this is a mossy fiber, which is the complete input, sensory input, except the olivocerebellar. So the mossy fiber is forming a glomerulus here with the granule cells and Golgi cells. And you can see the granule cell axon is entering into the molecular layer and divides like a T and forms the parallel fibers in the molecular layer. So in this way, see, one mossy fiber can stimulate many Purkinje cells, but here one climbing fiber is stimulating or influencing only the one Purkinje cell. And you can see the output is solely from the Purkinje cells. These are the output axons from the Purkinje cells, which are the afferents. These are the afferents. So mainly the afferent is mossy fibers and climbing fibers. All afferents are mossy fibers except olivocerebellar, which is the climbing fiber. So feed forward versus feed backward inhibition. So this is most important. If you see the climbing fibers, they facilitate Purkinje cells inhibition of deep cerebellar nuclei. The same climbing fibers also stimulate basal cells and stellate cells, which in turn exert their inhibitory function on Purkinje cells. So this is feed forward inhibition. So climbing fibers, it is actually stimulating Purkinje cells and it's also stimulating basket cells and stellate cells. So when this climbing fiber stimulates Purkinje cells, so this Purkinje cells actually inhibit deep cerebellar nuclei. At the same time, it is simultaneously stimulating basket cells and stellate cells. These will inhibit the Purkinje cells. So when they inhibit Purkinje cells, the inhibitory effect of Purkinje cells will be inhibited. So this is feed forward inhibition. Same way, mossy fibers also will do the feed forward inhibition. Like mossy fibers stimulate granule cells through parallel fibers stimulate Purkinje, Golgi, basket, and stellate cells. This is feed forward. But there is feed backward with the mossy. How it is happening? Inhibitory Golgi on facilitation suppresses excitation of the granule cells. This process of alteration of excitatory influence of granule cells is feed backward inhibition. Thank you.